So the focus of my lab is trying to understand how and why dying cells actually generate this type of extracellular vesicle called apoptotic bodies. And right from the start, I just want to clarify that apoptotic bodies are distinct from the large exophytes or large oncocytome that we've talked about today, because apoptotic bodies, by definition, should only be generated during apoptosis. And also, I just want to acknowledge that during apoptosis, other smaller EVs are also released, such as exosome-like ApoEVs. But based on our approaches, we don't actually study them, but hopefully we can study them in the future as well. So I would just like to start with a quote that from a very respected scientist in Australia. He said, "Nothing interesting actually occurs after back-to-back activation because the cell is already dead." As you can see, it's actually quite hard to get funding in Australia if they have this view, right? But to me, the most interesting part is when the cell has actually committed to undergo cell death. And the reason why I call this talk the last dance of a dying cell is because when cells undergo cell death, they undergo quite some remarkable morphological change that's actually quite beautiful in my point of view. So here's just a video, hopefully it will play, showing a monocyte undergoing apoptosis after UV radiation. And what you will see is that the cell will first undergo some cell rounding, eventually form this blebbing. And then it will actually form all this really thin string-like protrusion, which then become beaded and actually undergo fragmentation to generate what we call apoptotic bodies. Here's just another microscopic image showing that again, when the cells die, they form blebs, and the formation of this thin string-like protrusion that should push out the blebs to generate what we call apoptotic bodies. When I used to present this work, the first question that people will ask me, if you ask me nicely, is that, do you see this under in vivo condition? If you ask me not so nicely, it's that, is this an in vitro artifact? But we were really happy when we saw this paper from Sarah Kusin's lab from UVA, where they were interested in looking at how neural crest cells die and being cleared. And what they saw was when the cells actually die, they saw quite similar morphology as what we described in vitro, where the cells, shown in red here, will undergo blebbing and the formation of this protrusion, and then the protrusion undergo fragmentation to generate apoptotic bodies. So why do we care about this process? So I'm from the field of cell clearance, and we're particularly interested in how dying cells are rapidly removed by phagocytes. Because if dying cells are actually not removed efficiently, the dying cells, the membrane would become permeabilized, release intracellular contents that can cause inflammation. And there are a number of really cool mechanisms to ensure this process is actually very efficient. So for example, apoptotic cells can release what we call find me signals, such as nucleotides and also small EVs, that can then recruit phagocytes towards the site of cell death. And during apoptosis, dying cells can also undergo what I mentioned before, fragmentation to generate apoptotic bodies. And the exposure of what we call eat-me signals, such as phosphatidylserine, would then be recognized by phagocyte and trigger the cell clearance. And back then, it's not really well known how the apoptotic body formation are actually occurring. So I decided to establish a lab to just focus on this specific question. And what we're interested in is to try to develop methodologies to study how cells generate apoptotic bodies. In particular, what are the molecular mechanisms of this process? Obviously, try to understand what's the function of this process. And one of our major goals is to identify drugs that can actually manipulate this process in certain settings. So I'm very lucky to have a fantastic team working on this work together. And the work that I present today is done predominantly by a very talented postdoc, Dr. Carfan. So just a bit of background on the mechanism of how cells actually generate apoptotic bodies. So it's been known for a very long time, for more than 30 years, that cells, when they undergo apoptosis, they undergo membrane blebbing. And if initially thought to be a random process, they would generate apoptotic bodies, which then be cleared by phagocyte. A few years ago, we discovered that after the membrane blebbing step, again similar to ixophar, that the cells actually form protrusion to actually push out the blebs to form apoptotic bodies. And they come in two different flavors. One is a very thin string-like protrusion that happens after the blebbing, push the blebs apart to form apoptotic bodies. And then there's this other type of protrusion generated by monocyte, whereby they form this protrusion that become beaded and then undergo fragmentation to generate apoptotic bodies. And based on this kind of observation, we come with this conceptual framework at that time, that thinking that how cells actually undergo disassembly to generate apoptotic bodies is regulated by three sequential steps. Step one is membrane blebbing, and then the formation of the protrusions, and then final separation into distinct apoptotic bodies. And obviously, we're really interested in what are the molecular regulators of this process. So it's been known for quite a long time now that ROCK1 kinase is actually being activated during apoptosis by caspase, which then drive ectomyosin contraction to form membrane blebbing. And recently, we have also demonstrated that this is also quite essential for the formation of apoptotic bodies. Hold on. And then a few years ago, we also identified actually a negative regulator of this process called pannexin-1, which is again a caspase cleaved protein. And what happens is, if you block pannexin-1, cells are more likely to form this protrusion and leading to more likely the chance of forming apoptotic bodies. And recently, we've also identified a transmembrane receptor called plexin B2 that plays a key role in just the formation of the beta Apoptopodia that I mentioned before for monocytes. And part of the talk today, I'll focus on another new regulator of this particular step. So how do we actually discover a new regulator of this process? Initially, we took a number of different approaches. So we did drug screen in collaboration with the Ravichandran laboratory at UVA. We also did proteomic analysis of the VC apoptotic bodies with the Methylvinen lab at La Trobe. We also did a genome-wide CRISPR screen with the Harrow lab at Weihai. But what we'll focus on today is the drug screen. So the system that we use is actually quite simple. So we've got some human surrogate T cells, induce them to undergo apoptosis by anti-Fas, which then will undergo blebbing. And a lot of them would then form apoptotic bodies. And then what we did next was establish a pretty robust protocol by flow cytometry that can measure the amount of apoptotic bodies relative to apoptotic cells. And then we did a drug screen using the LOPAC library, which contains about 1,300 drugs, with most of them with a non-molecular target. So therefore, if any of those drugs actually affected the level of apoptotic bodies, that will give us a clue of what potential regulator of this process. And the one that, when I gave this data to my postdoc Carr, he decided to focus on the particular drug called mebrafidil, MBF, which is a calcium channel inhibitor. So as I described to you before, when cells die, they undergo blebbing, formation of the protrusion to separate the blebs to form apoptotic bodies. In the presence of mebrafidil, it can block the formation of bodies, apoptotic bodies, as measured by flow cytometry. And because most of our assay, we also monitor how cells die by time-lapse microscopy. So therefore, we can actually pinpoint that mebrafidil only affected the protrusion formation step, but not the blebbing step. Because mebrafidil is known to be a T-type calcium channel inhibitors, we look at other T-type calcium channel inhibitors, and they all inhibited the formation of apoptotic bodies. Whereas when we use inhibitors to like N-type or L-type or store-operated calcium channel, it doesn't have, that doesn't do anything in this scenario. So we think that T-type calcium channel is another new regulator of how apoptotic bodies are being formed. So T-type calcium channel, the transmembrane channel, activated by membrane depolarization, meaning increase in membrane potential, which then allows calcium influx into the cell. And there are three isoforms, CAF3.1, 3.2, and 3.3, encoded by those genes. And CAF3.1 and CAF3.2 are expressed in both excitable and non-excitable cells, whereas CAF3.3 is expressed only in the brain tissues. I guess lucky for us with the human surrogate T cells that we're using, they pretty much only express CAF3.2. And when we knock down CAF3.2, we can see a clear reduction in the formation of apoptotic bodies and also the protrusion formation step, suggesting that CAF3.2 is a key positive regulator of apoptotic body formation. And if calcium channels is involved, I guess it's no surprise that calcium flux could also play a role. So here it's quite interesting. After you induce apoptosis, and then prior to cells actually undergoing fragmentation, in the black line, cells actually undergo quite a lot of fluxes in this process. And this can be blocked by calcium flux, and this can be blocked by inner MBF, or simply chelating the extracellular calcium by EGTA. And in fact, just having EGTA can actually block the formation of apoptotic bodies. So all this data coming together suggesting that calcium influx through CAF3.2 is important for the cells to generate apoptotic bodies. I'll come back to CAF3.2 a bit later. But right now, I just want to discuss a little bit more about the function of apoptotic bodies formation. So a few years ago, we described that by manipulating the regulators I've described so far, that the formation of apoptotic bodies is actually important for cell clearance. So the analogy that I like to give is that, imagine you're eating a piece of steak. You need to cut them up into smaller pieces to eat them right. So it's similar for apoptotic cells. If they can actually fragment and break apart into smaller pieces, it makes it easier for certain phagocytes to actually engulf them. But in these studies, we pretty much just use in vitro system. So right now, we're particularly interested in trying to develop some in vivo assays or in vivo tools to try and study this process. I'm just going to describe to you briefly about this. So as I mentioned before, the two key regulators of this process, ROCK1 and pannexin-1, they're both activated in apoptosis by caspase. And then in collaboration with Michael Olsen, he's very generous and shared with us the ROCK caspase resistance mice, meaning that ROCK1, the caspase cleavage site in ROCK1 is mutated, meaning that ROCK1 won't get activated during apoptosis. And therefore, cells derived from these mice won't undergo membrane blebbing, and hence will form less apoptotic bodies. We've also developed our own new strand of mice, where we call pannexin caspase resistance mice. Similar to the ROCK mice, again, the pannexin, the caspase cleavage site on the pannexin is mutated, so therefore, pannexin-1 won't get activated during apoptosis. By removing this negative regulator, the cells are more likely to form the protrusions, and then more likely to form apoptotic vesicles. So we've confirmed that these mice do have those phenotypes, and right now we're screening these mice for a variety of different diseases to see whether it has a particular phenotype. And hopefully I can present this, or someone in my lab can present this in another meeting. But what I do want to focus on today is the role of apoptotic bodies in intercellular communication. Right. So initially, we were interested in this in the context of influenza virus infection, because because at that time we were interested in how monocytes undergoing disassembly to form apoptotic bodies, and it's also well known that when monocytes are infected with influenza virus, it can undergo apoptosis. And what we demonstrate initially is that similar to UV-treated cells to induce apoptosis, when we induce monocytes to undergo infect them with influenza, and when they undergo apoptosis, they'll again generate the beta protrusion that I mentioned before. But what's important that we discovered in this work in collaboration with Wei Chen and Latrobe is that the apoptotic vesicle actually contain virions. And when we isolated the apoptotic bodies by FACS sorting, and we give them to other cells or give them to mice, it will actually propagate the infection, suggesting that apoptotic bodies can actually traffic viral particles between the dying cells and healthy cells. And obviously, when SARS came along, Carr, again from my lab, was quite interested in this process to see whether this is similar happening in the setting of SARS. And we did this in collaboration with Anna Cousins from Weihai. And what we initially did was we infected some viral epithelial cells with SARS-CoV-2, which is well known to induce apoptosis in these cells, and then also, or with our control apoptosis inducer APT737. In both settings, the cells will undergo disassembly to generate apoptotic bodies. But what's important to note here is that only in SARS, obviously SARS infected cells that are undergoing apoptosis, the apoptotic vesicle will actually contain viral protein, viral RNA, and also viable virions. We also observed similar things under in vivo condition, where we infected some aged mice with a most adapted strain of SARS-CoV-2, and 3 days post-infection, when we look at the bowel, we can also detect apoptotic bodies. And next thing that we did was we isolated those apoptotic bodies by differential centrifugation, and then we give them to 2 different cells types. One is to give them back to viral cells, and the other one to give them to a monocyte-derived macrophage. I guess it's no surprise that because the apoptotic bodies actually contain virions, when we give them to viral cells, it can actually cause them to undergo cell death because they become infected and actually die. But what's actually very interesting regarding the monocyte-derived macrophage is that under in vitro condition, it's actually very hard to infect macrophage with SARS-CoV-2 because it's known that they express a low level of ACE2 receptor. But what's interesting is that when we incubate the apoptotic bodies with the macrophage, a small proportion of them will become infected, and then it will also release a variety of different pro-inflammatory cytokines, such as IL-1 beta. So based on this, we think that the formation of apoptotic bodies under infection setting could be actually a bad thing, because the apoptotic bodies can actually traffic the virus to another cells to cause cell death and inflammatory response. So therefore, we ask the next question: what happens when we actually block this process? I guess no surprise, we decided to target the Ca3.2. So in virial cells, they do also express the protein I mentioned, the calcium channel that I mentioned before, Ca3.2. In the presence of mebrafidil, it can also block the formation of apoptotic bodies generated by SARS-infected cells. I just want to mention here that mebrafidil does not affect how SARS infect the cells or the ability of the cells to die. It's just the apoptotic bodies formation step. And one of the experiments that we did was that we first infected virial cells with SARS-CoV-2, and then we treat the cells in the absence of mebrafidil. And then we collect the conditioned media and then give them back to virial cells. In this case, because the conditioned media actually contains apoptotic bodies and also live virions, it will actually cause the virial cells to die. But then in the presence of mebrafidil, it can partially rescue that. Similarly, in the macrophage setting, when we treat the cells with mebrafidil, it can also block the release of a variety of pro-inflammatory cytokines, such as IL-1 beta. So collectively, based on this data, we think that when cells are infected with SARS-CoV-2 and when they undergo apoptosis, the apoptotic bodies generated under that setting contains viable virions. And I should have time to describe today, but I don't have the data. But if you're interested, I can show it to you a bit later, that when cells the apoptotic vesicles, through an efferocytosis-dependent mechanism, can traffic the virions to epithelial cells and also other macrophages and cause them to die and actually elicit a pro-inflammatory response. So therefore, we think that potentially blocking the formation of apoptotic bodies or maybe actually targeting the efferocytosis step could be a way to limit this particular pathway of trafficking of SARS-CoV-2. So in summary, hopefully I convinced you today that how cells actually undergo disassembly and generate apoptotic bodies is regulated by 3 distinct sequential steps: the blebbing, the protrusion formation step, and the final separation. And we've identified a number of regulators of this process. And the formation of apoptotic bodies is key to enhance the clearance of dying cells. And under the setting of infection, the apoptotic bodies may contain virions and hence traffic to other cells through this process. I guess one of the long-term goals of my lab is trying to identify inhibitors of this process. And we have identified a few, and the hope is potentially under certain setting, blocking the formation of apoptotic bodies could be beneficial. Another one of my favorite topics is actually to try to enhance the cells to actually undergo disassembly, because this may be beneficial to promote the clearance of dying cells and could be important for certain chronic inflammatory disease. And we can do this by targeting this channel called pannexin-1, which is a negative regulator. And we've also identified a few inhibitors of this process. So finally, I'd like to thank my group, which in particular Ka Feng, who did most of the work that I described today, funding bodies, as well as a variety of collaborators, in particular Anna Cousins from WeHi. Thank you.
Thank you so much for this exciting talk. We have time for a few questions. Maybe I start. I'm not sure if I listened properly, but do dying cells also release EVs?
Sorry?
Do dying cells also release EVs?
EVs, the small, you mean small EVs?
Yeah.
Yep, absolutely. So,
or large, whatever.
Sorry?
Or large.
Or large, yeah. Well, that's what we show. The large EV, they release large apoptotic bodies, but they also release small exosome-like vesicle. But we don't study them as much. But I think that's another interesting point that we will be looking into, because whether even the formation of the smaller EVs use similar mechanism, because we've already got all the tools already.
Okay, so we have our first question there.
Hi, great talk, by the
Can
way.
go very
okay.
and introduce yourself, please.
I'm from France. My name is Liam. Great talk, by the way. I was interested with the increase in the LPS. If you think it's due to something on the surface of the AB, or is it the contents? Because there could be activation of the TLR receptors. And so I was wondering if the surface markers of the ABs could be stimulating this increase.
Oh, I haven't got used LPS in any of, but it's exposure of phosphatidylserine.
EL1 beta, sorry. Yeah, so I was wondering if the increase in the EL1 beta, you think it's due to the contents of the AB, or if it's due to a marker?
Yeah, so I think at this stage we think it's definitely due to the infection of those cells. Because it's true, the cells that were infected with SARS-CoV-2, the bodies itself could contain pro-inflammatory cytokine. We haven't looked into this too much. But then what the critical experiment that we did was that when we give them to the phagocytes, we actually sort out cells that actually undergoing engulfment. In those settings, the content is already degraded. But then it's those cells that become infected, actually. But it's not productive infection, but they still become infected, and that's why they release the cytokine.
Okay, thank you.
Clotilde? Oh, sorry. Ladies
Go ahead.
first. Okay.
Thank you. So, Clotilde Terry. I'm just wondering if the cells, the EVs, I guess the ApoEVs are very heterogeneous, like other EVs. And I was wondering whether the virus were present in all of the released EVs, or whether maybe there could be some of EVs that would carry, for instance, ACE2, the receptor for the virus, as it's been shown by us and others that ACE2-bearing EVs could inhibit further infection. So I was wondering whether there could be contradictory effects of the ApoBDs released, some like transferring the virus, as you show, but some others preventing infection, reinfection of other cells.
No.
Did you explore the heterogeneity or not?
No, we haven't yet explored the heterogeneity, but I totally agree with the heterogeneity because we know that a lot of the intracellular contents are very different between apoptotic bodies that we isolate. A proportion of them would, like in the extrovert, contain certain intracellular content as well as surface material. So it is possible that could happen, and then also something that has been us wondering is that the size of the EV could also be critical in that sense, because we think that the uptake mechanism, not all of the vesicle uptake would be done in the same particular way, and only maybe particular size that can actually regulate this process as well. But definitely, if they may carry ACE2, and then again could be what they call it, like a decoy mechanism in a way.
Thanks.
Yeah.
Hello, Ivan.
No,
Sorry.
I'm sorry. I think I promised the group on the left here first, and then we'll get back to you.
Thank you. Thank you. Great talk. Alan from Boston Children's Hospital. So I was wondering that if you block the apoptotic formation with the T-type calcium inhibitor, do the cells still die? Do they still die from apoptosis, or maybe it shifts to other pyroptosis or other types of cell death?
Yeah, absolutely. So we've done all those control experiments. So making sure that, first of all, when we block them, it doesn't affect the level of apoptosis. Because of our assay, we also make sure that it doesn't go necrosis. And then we also done a variety of caspase activity assays, blocking assays to demonstrate that that would be the case. So it's not like we're driving necrosis. That's the biggest worry, because, for example, if you use drug, if you make them undergo necrosis or apoptosis, obviously the response is different. But,
So does it prolong their cell life using this type of inhibitor?
Prolong the cell life,
Like,
as in?
Yeah, as in like you induce apoptosis first, but then you block this calcium channel. Does it prolong your THP1s, or does it prolong your T cells?
No. Viability-wise, no. But the stability-wise of the vesicle, maybe. That's another story. Yes.
Sorry, a follow-up question. So with your beaded Apoptodia and also your apoptotic bodies, do they have a preference in like a membrane polarization, or where or when one forms or the other forms?
That's a question that we struggling to address, and it's quite interesting because they form everywhere. So it's not like one half the cell will form the protrusion when we see it's surrounding the cells forming. So we don't know why, but one thing that we do know though is that it's synchronous. So therefore, either they form at the same time.
Maybe we can talk later, because I have a video to show you on neutrophil death that we see the same thing, actually. And we have a sequential thing. I'll show you later in coffee break.
Yeah.
Okay, thank you. So networking is already happening. And you are next, and then Matthias afterwards. And then we go to our featured abstract.
Okay, thank you so much. Just a quick question. Did you try to do the same experiments using primary cells? Like, for example, monocytes, macrophages, and to check if this mechanism that you are trying to explore is the same thing in the donors? For example, the apoptotic, the apoptosis thing, all the things with primary cells?
Yes. So this is more in general. So for example, all the regulators that we use, all the inhibitors that we use, and even the morphology that we use, do we use primary cells? Yes, we do. So basically, for example, Pannexin 1, ROC1, plexin B2, all of them, there's an aspect in the paper that we have. We did use primary cells for that work. And then obviously now, we have, trying to manipulate those regulators in mice, right? So for example, we're looking in, whether it's, not meth, it's not, but, primary thymocytes, max from there, and we saw very similar phenotypes.
Thank you.
Okay, last but not least.
Yes. Hello, I'm Matías Ostrowski from University of Buenos Aires. Because I guess that when you have the apoptotic cell, the infected apoptotic cell, you have free virus and then the virus contained in the apoptotic body. So my question is, if you compare the infectivity of the free virus and the virus inside the apoptotic body, and related to that, how does it relate to the process of apoptotic mimicry that many viruses like SARS-CoV-2 also use to increase infectivity?
Yeah, I mean, we haven't looked at the concept of apoptotic mimicry yet, but what's interesting with the macrophage experiment that I show you is that Carl was being very careful in terms of the amount of virus that he used compared to the amount of so-called virus in the apoptotic bodies. And prior to the experiment, he would demonstrate that the level is similar between them for cells that, like viral cells, for example, when they do the TCR-ID50 assay. But then, when the same amount was then added to the macrophage, it's only the apoptotic bodies that cause infection. So we'll be very careful with the comparison as well. But in that case, it's only the apoptotic bodies that actually cause a small amount of infection.
And do you have any idea of why?
Idea of why? We know it's an efferocytosis-dependent mechanism. So therefore, it's been known for a while that, for example, HIV and other intracellular bacteria can, through that mechanism, to infect cells. But what we don't know, and how long it will take to find out, is after engulfment, how does it escape? That's something that we don't know.
Okay, thank you.
Okay, thank you so much.
Thanks again, Ivan, Poon, for this wonderful presentation.